Could living beings assemble from silicon backbones, metal lattices, or even solvents like liquid ammonia, thriving in worlds utterly alien to Earth's wet, carbon-rich biology? In this hypothesis, we imagine life not built on proteins and DNA, but on entirely different chemistries, organisms with silicon skeletons, flexing in methane seas, metal oxide frameworks conducting electrons instead of blood, or ammonia-soaked vesicles shuttling nutrients in frigid clouds. Each scenario stretches our definition of alive, asking whether metabolism, growth, and reproduction could emerge from bonds and solvents we rarely consider hospitable. What makes carbon the superstar of Earth life? Carbon's unique ability to form four stable bonds under biological conditions lets it weave chains, rings, and complex 3D architectures crucial for proteins, fats, carbohydrates, and nucleic acids. Its bonds are strong enough to hold life's machinery together, yet breakable enough to allow enzymes to rearrange molecular pieces. And because carbon compounds dissolve in water but resist random degradation, they balance stability and flexibility in a way few other elements can match. But could chemistry find other routes? Let's explore some daring alternatives. Silicon, just below carbon on the periodic table, can also bond in fours, but its bonds tend toward brittle, rock-like networks rather than supple chains. In cold, methane-rich oceans like Titan's, though, silicon-oxygen polymers might flex into membrane-like shells, while hyperbranched silicon dendrimers mimic enzyme pockets. Ammonia itself, liquid at much lower temperatures, could dissolve ammonia polymers that fold into catalytic shapes. Hydrocarbon lakes might host azotosome membranes, nitrogen-rich analogs of our lipid bilayers, allowing creatures to sip methane like we drink water. Some Earth microbes even swap phosphorus for arsenic in DNA, hinting that unusual elements can join the dance of life. Boron hydrides and polyphosphates present further frameworks for information storage, while metal organic clusters on mineral surfaces could catalyze reactions without ever leaving the rock. In the wildest visions, charged dust storms or plasma filaments in gas giants form dynamic lattices, processing information through standing electromagnetic waves. How would these beings eat and breathe? Rather than inhale oxygen, silicon-based life might shuttle electrons through conductive backbones, breathing charged particles to power metabolism. Ammonia solvent organisms could exploit hydrazine or methane redox couples, harnessing chemical gradients like miniature fuel cells. Metal organism hybrids might oxidize metal sulfides or conduct energy via nanowire networks, turning inorganic substrates into living batteries. And on stormy gas giants, coherent plasma structures could harvest radio waves or magnetic fluctuations, converting energy directly from their turbulent skies. Could we detect such life? Yes, if we know what to look for. Unusual atmospheric gases like silane CH4, phosphine PH3, or exotic hydrocarbons might signal silicon or ammonia chemistries at work. Surface patterns of repeating, non-random mineral or ice structures could betray colony growth, while spectral flashes from metal-catalyzed reactions might glow in infrared or ultraviolet bands. On icy moons, arrays of organic or inorganic rings in the ice, unmoved by geology, would scream bio rather than geology. The non-carbon-based life hypothesis suggests that alternative chemistries such as silicon or metal-centered molecules could form the basis of living systems. Unlike carbon, silicon atoms can also form four bonds, but tend to create stronger, less flexible networks and unstable oxides, while metal-based life would rely on coordination complexes and redox cycles centered on transition metals rather than organic polymers. Such exotic biochemistries challenge the carbon-centric paradigm by proposing that life's essentials, information storage, metabolism, and membranes, might arise from entirely different building blocks, theoretical silicon analogs of DNA. Silifuranose backbones with nucleobase-like side groups have been modeled for information replication. much like imagining houses built not from brick and mortar but from interlocking metal frames and magnetic joints. Carbon's versatility arises from its ability to form stable, single, double, and triple bonds, enabling complex, flexible macromolecules, while silicon forms strong silicon-oxygen bonds but weaker silicon-silicon linkages and less stable silicon-hydrogen bonds, limiting its polymer diversity in aqueous environments. Metal-based biochemistry would center on coordination of ligands to metal ions, creating catalytic centers akin to enzyme active sites but lacking long, information-rich backbones, fundamentally differing from terrestrial organisms. Iron-sulfur clusters in Earth life hint at primitive metal-based catalysis, but no known organism uses metals as informational polymers, much like comparing a flexible leather belt made of carbon polymers with a rigid chain-link fence of silicon network. At the atomic level, silicon's larger size and lower electronegativity produce bond angles and energies that favor three-dimensional networks over linear chains, and in water, silicon compounds hydrolyze readily to form silicic acid, undermining the stability of silicon backbone molecule. Metals coordinate ligands via variable oxidation states, offering catalytic versatility but little capacity for stable sequence-based information storage, making it difficult to assemble silicon or metal equivalents of proteins and nucleic acids. Silicon polymers analogous to polyethylene degrade rapidly in water, much like trying to build a rope out of slippery bar magnets. They stick unpredictably and dissolve in rain. Silicon-based life would require non-aqueous solvents, such as liquid ammonia or sulfuric acid, where silicon bonds are more stable and information might be stored in polypeptoid backbones with metal chelated side groups, or in crystal lattices with defect patterns, while metabolic reactions could leverage metal redox cycles, cycling iron, copper, or titanium centers to generate energy gradients. These alternative mechanisms illustrate how different solvents and coordination chemistry could substitute for water and carbon polymers in supporting life's core processes. Hypothetical life in Venus's sulfuric acid clouds could use sulfosilicon polymers stable only at low pH, much like using a different currency in another country. Everything works similarly, but the underlying tokens and exchange rules change. Extreme environments such as the sulfuric acid clouds of Venus, supercritical CO2 on exoplanets, or molten metal lakes could favor non-carbon chemistries that thrive where water-based life cannot, and natural selection would favor molecular systems that remain stable, replicate, and catalyze reactions under such harsh conditions. Silicon or metal life likely exploits temperature, pressure, or solvent niches inaccessible to Earth organisms, and environmental constraints thus guide which elemental frameworks can support life. Titan's cold methane lakes, for instance, might host silicon-based lipid analogs that remain liquid where water freezes. Much like certain plants, evolving antifreeze proteins to survive sub-zero climates where others perish. Forming stable silicone polymers requires surpassing energy thresholds in non-aqueous solvents. Below that threshold, monomers decompose rather than link, and metal coordination complexes show catalytic activity only when ligand field strengths exceed critical values. Otherwise, they remain inert. These nonlinear chemical thresholds create sharp viability windows for exotic life chemistries, and understanding them is key to assessing feasibility. Laboratory synthesis of long siloxane chains demands temperatures above 300 degrees Celsius in dry conditions, much like trying to bake bread at too low a temperature. The dough never rises and remains raw. Discovering non-carbon life would revolutionize our understanding of biology, expanding the definition of habitable worlds, while synthetic biology aims to create orthogonal life forms, such as silicon backbone genetic systems. To test these possibilities in the lab, potentially revealing fundamental principles of life's emergence and adaptability, The search for life beyond Earth must therefore include signatures of non-carbon chemistries and metal redox cycles. Researchers are developing nucleic acid analogs with XNA xenonucleic acids that use non-ribose backbones to test alternative information systems, much like programming robots with entirely new hardware architectures to explore the limits of machine intelligence. Spectroscopic signals of silicon or metal-based biomolecules may overlap with abiotic mineral signatures, complicating remote sensing, and modeling such life requires integrating geochemistry, non-aqueous solvent physics, and unconventional reaction networks, a formidable interdisciplinary challenge. Laboratory analogs must recreate extreme conditions of pressure, temperature, and solvent composition to validate these hypotheses. Detecting silane-based lipids in Titan analog experiments, for instance, struggles against background silicate dust signals, much like seeking a specific radio station in a city filled with static and overlapping broadcasts. Advances in high-pressure microfluidics, cryogenic reactor design, and quantum chemistry simulations will refine our predictions of silicon and metal life viability, and space missions targeting Venus's atmosphere, Titan's lakes, or exoplanet spectra should include instruments tuned to non-carbon biosignatures. Synthetic biology may soon yield proof-of-principle organisms with novel backbones, illuminating life's chemical flexibility. Ultimately, confirming non-carbon-based life would expand the tapestry of biology and redefine our place in the cosmos. Planned probes to Venus aim to sample cloud droplets for silicon-bearing organics, alongside sulfur compounds. Much like dispatching deep-sea submersibles to hydrothermal vents once thought inhospitable, only to discover thriving ecosystems in the dark.